we conclude this chapter, the conversation about the anatomy and basic physiology of the lymphatic system. It's really fairly straightforward. So what essentially happens is when the blood, there's a filtration and reabsorption. Filtration happens at the arterial end of the capillary beds, and reabsorption happens at the venular end. So fluid leaves at the arterial end, but not all of the fluid is returned on the venular end, so that excessive fluid that leaked from the blood are collected into the lymphatic system. Here in this image you can see the blood capillary bed and lymphatic capillaries next to the blood capillaries. Okay, the order is it's a lymphatic capillary that collects the lymph, and then lymph is drained into the collecting vessels. From collecting vessels through lymph nodes into the lymphatic trunks and ducts. As we go from capillaries through collecting vessels through trunks and ducts, the diameter of the lymphatic vessels progressively increases. Okay, so the role of the lymph nodes is to filter lymph. They have loose reticular connective tissue that serves as some kind of a mesh in which multiple immune cells are embedded, cells like macrophages, dendritic cells, and T and B lymphocytes. Along with lymph nodes, there are other organs of lymphatic system. So you see spleen, thymus, tonsils, and Peyer's patches. Peyer's patches are found in the gut. There is a diffuse immune tissue found in many various organs. For instance, lamina propria, there are diffuse immune cells, not organized in any particular structures. Now, the system is one-way system. The direction of a lymphatic flow essentially is from tissues towards the heart eventually. Okay? Now, let's take a look at organization of the small lymphatic vessels. This is a snapshot of the lymphatic capillary. Okay, you can see that it is anchored by these filaments to the connective tissue, and these individual slats are individual endothelial cells, lymphatic endothelial cells. Now, if you take a closer look right about here, you will see one cell forming a valve with another cell. When hydrostatic pressure in the tissues increases, this hydrostatic pressure pushes on that valve, pushing lymph inside of the capillary, and the lymph, that's how fluid enters the lymphatic system. Does that make sense? So lymphatic vessels, lymphatic capillaries are found almost everywhere, except for several locations, including teeth, bone marrow, bones, and previously, sorry I did not upgrade this lecture up until 2 years ago, central nervous system. If you think that there are no discoveries in anatomy 2 years ago, it was proven wrong when lymphatic, very branched and extensive lymphatic system was found in the brain. Turns out we do have lymphatic vessels there. A special kind of lymphatic capillaries, as you know, lacteals, they found in mucosa of intestines, and they collect fat, collect lipids, and deliver them to the blood. Now, lymphatic capillaries merge, forming collecting vessels. Collecting lymphatic vessel is kind of similar to the vein. It has valves, a bit thinner walls, and more anastomosis, so it makes more branched network. So where are you going to find lymphatic vessels? They usually run along the veins or deep arteries. And you will see why, considering how lymph is transported, running along the artery makes perfect sense. So this collecting lymphatic vessels, shown in this image, merge further together, forming thoracic duct. Okay? Thoracic duct collects lymph from the bottom part of the body and from the left arm and left side of the thorax and the head. So that area that is bound by the red color is the area of lymph collection by thoracic duct. Now, right lymphatic duct, shown right here, collects the lymph from the right arm and right side of the head and thorax. That would be like this. Does that make sense? So you see that thoracic duct collects lymph from much larger area of the body. It kind of matches the branching of the arteries from the sort of matches branching the arteries from the aorta. When you have a brachiocephalic trunk that supplies the blood to the right arm and right side of the thorax and the head, and you have separate aorta, left subclavian and left carotid arteries that deliver the blood to the rest of the body. Does that make sense? Now, where do these ducts deliver the blood? First of all, they deliver it at the converging point between the internal jugular artery and internal jugular vein and subclavian vein. I'm sorry. Okay, so you see here, that's internal jugular, that's subclavian, that's where the ducts empty. Good? Now, ducts are formed by multiple trunks, and you see main trunks on the right picture. Intestinal trunk that collects it from intestine lumbar trunks, from the lower limbs. Bronchomediastinal trunk that collects lymph from the thorax and the right arm. Jugular trunk and subclavian trunk, also right arm and right part of the head, and same set of trunks on the left side. Okay, so basically the idea is, as I mentioned before, trunks merge into the ducts, deliver the lymph to the ducts, ducts empty the lymph into the subclavian veins. Does that make sense? Now, when lymph is collected into the capillaries and then enters the collecting vessels, you have to understand that's the plumbing system, just as the cardiovascular system is. The main difference between the lymphatic system and cardiovascular system is that lymphatic system lacks the central pump. Is that clear? So there has to be some kind of a force that allows the lymph to move from collecting vessels into trunks and then into ducts. And these forces are very similar to the forces that move blood in the veins. Skeletal pump or muscle pump and respiratory pump. That's probably the most important way the lymph is propelled through the collecting vessels. I mentioned before that collecting vessels and trunks often run next to the deep arteries. It turns out that pulsatile nature of the arteries also facilitates the lymphatic propulsion through the collecting vessels and trunks. Lastly, smooth muscle cells found in the walls of the collecting vessels, lymphatic trunks, can contract, weakly contract, to add other dimension to the propulsion forces of the lymph. That makes sense? Just as in the veins, lymphatic vessels have valves that prevent the backflow of the lymph. So lymph flows through the lymphatic vessels and eventually ends up in the lymph node, where it will be filtered through the mesh of reticular fibers. Reticular fibers are produced by the reticular cells. It's basically specialized types of fibroblasts. Okay? And in that mesh, you're going to find lymphocytes, B and T cells, that we just discussed, and the macrophages. So the role of macrophages is fairly straightforward. Does that make sense? They just phagocytose whatever is in the lymph. Think of the lymph node as the small water treatment station on the way of the lymph to the blood. Dendritic cells found in the lymph nodes will also phagocytose pathogens, but with slightly different purpose. They will phagocytose pathogens to present the antigens to T and B cells. Remember that mature T cells and mature B cells, after their maturation respectively in thymus and bone marrow, will migrate into the lymphatic tissue and will seed lymph nodes and spleen, because that's where they're going to encounter antigen. There are two types of lymphatic tissue: diffuse and follicular. So diffuse lymphoid or lymphatic tissue can be found everywhere, quite literally everywhere. If you would look in the connective tissue, for instance, you would find diffuse immune cells in that connective tissue. Now, follicular lymphatic tissue, follicular lymphoid tissue, found in lymph nodes, Peyer's patches. Okay? And in lymph nodes, Peyer's patches, tonsils, you're going to find an accumulation of B cells and T cells that are exposed to the antigen and are proliferating and differentiating in response to that exposure. So let's take a look at the structure of the lymph node. So lymph node is shaped like a bean, okay? And in terms of the size, they're pretty small, about the size of a small bean actually. There are certain areas in the body which have more lymph nodes than others. The most notable are areas under the armpits, so-called axillary lymph nodes, in the neck, so-called cervical lymph nodes, and in the groin, so-called inguinal lymph nodes. Lymph nodes that may be less palpable, not shown on this picture, are popliteal lymph nodes, found in the back of the knees, and the huge accumulation of lymph nodes can be found in the mesentery, in your gut. So as you can see, lymph nodes are located either near the large vessels or at the potential sites where pathogens can enter the body. Cervical lymph nodes near the opening of the respiratory tract, inguinal lymph nodes close to the reproductive organs, and mesenteric lymph nodes close to the digestive organs. So, three main types of cells, functional types of cells, found in the lymph nodes are macrophages and T and B lymphocytes. Lymph node is enclosed in the fibrous capsule, and fibrous trabeculae break the inner space of the lymph node into separate compartments. As many other parenchymatous organs like spleen or thymus or kidney, lymph node has outer and inner layer. Outer layer is called cortex, and inner layer is called medulla. In the outer layer, you're going to find follicles. Okay? With the germinal centers, that's the germinal center of the follicle, where B cells differentiate and proliferate. Okay? There are several stages of B cell development, and in fact, the initial stage of B cell development in the lymph node is called a germinal center B cell. So it's a B cell that have not become the plasma cell yet, but on its way there. Okay, around the follicles, dendritic cells can be found. Remember, dendritic cells are necessary for the antigen presentation to T cells. And that's what they do. They present antigen to T cells found in the deep cortex and in the medulla. Now in the medulla, in this medullary cords, you can find B cells, T cells, and finally plasma cells. If you look at the progression, now look, focus on the progression. Lymph, let's take a look at the lymph flow. Lymph enters the lymph node and encounters which layer? The cortex. B cells and T cells that are about to start developing. So that's how they get exposed to the pathogen. Does that make sense? And then, as they get exposed and get activated, fully functional B cells, T cells, and result of B cell differentiation, plasma cells, will be found in the deeper medulla, where they're going to be entering the blood to produce, antibodies, or circulation will deliver to the sites of infection. Does that make sense? Now, you see that there are lymphatic vessels getting into the lymph node and leaving it. And the names for these should be pretty familiar for you. The ones that enter the lymphatic vessels are called afferent, meaning coming in. And the ones that leave lymphatic vessels are called efferent, lymphatic vessels coming out. How many afferent lymphatic vessels do you see there? 5. 1, 2, 3, 4, 5. How many efferent lymphatic vessels? Only 2. So you have a bathtub with 5 faucets and 2 drainages. What's going to happen with the water level in that bathtub? Huh? It's going to rise, it's going to go up, right? Does that make sense? So the idea is that more afferent vessels and less efferent vessels make the flow in the lymph node, flow through the lymph node, is somewhat stagnant. So lymph stays in the lymph node for some period of time that allows the successful activation of B and T cells. Does that make sense? More afferent vessels means that lymph will stay in the lymph node. Make sense? We're clear. Now, another lymphoid organ is the spleen. It's the largest one. Blood is supplied through the splenic artery, drained by splenic vein. Spleen is also bean-shaped, large bean-shaped. Okay? So this large, okay, I have to take my words a bit back. If you actually would look at the spleen, it's not so much bean-shaped. Do you know what the fluke is? Like the helmet, The worm, fluke, no. Okay, imagine it's a tape. Spleen looks more like a tape, healthy spleen. You see what I'm saying? So it's a fairly short and wide tape, because when we say bean, we actually, can you imagine bean now? Okay, take a bean, flatten it out, stretch it out. You're going to get the spleen. Does that make sense? Now, size-wise, it's, pretty large. It's not bean-shaped. It's not the size of a bean. Okay? Size-wise, it's actually comparable. Length-wise, it's comparable to the stomach. Okay? So, the spleen plays the role in lymphocyte proliferation, accumulation of white blood cells, and also immune surveillance. So, let's break down the functions step by step. First of all, a lot of stuff is getting broken down in spleen, such as red blood cells, white blood cells, and platelets. So spleen recycles old blood cells. Second, when red blood cells get broken down, iron is released. That iron is stored partially in the spleen. Third, it stores platelets and monocytes. It was actually shown that in response to thrombocytopenia, spleen can release platelets right away. And in response to infection, it will release monocytes. Moreover, spleen will be the site of lymphocyte production, white blood cell production, along with bone marrow in case of infection. Does that make sense? Now, you may have heard the term splenomegaly. Have you? So splenomegaly is spleen enlargement that often can be found in chronic, long-term, severe chronic infections. When spleen has to produce large amounts of white blood cells, the spleen will get enlarged. Splenomegaly can also be observed in patients with various types of lymphoproliferative diseases, meaning lymphomas. Many patients with lymphomas have their spleen removed because spleen keeps producing defective white blood cells. Consider the function of the spleen. Yes? No.
But, so when you're sick, the reason why your glands are swollen is because of the high production of white blood cells?
Yes, exactly. It's not a dumb question. It's a great question. When you're sick, moreover, depends on how you're sick. If you are respiratory sick, nobody's going to look at your inguinal lymph nodes. They will look at your cervical lymph nodes, see enlargement. Okay, you have some kind of lymphadenitis here, local inflammatory process, so local response. Does that make sense? There is a condition known as lymphogranuloma venereum in response to long-term chronic, like really untreated old STD. People will have swollen inguinal lymph nodes, and they will be not bean-shaped. I mean, they will be bean-shaped. They will be like this large. So that's really noticeable, okay? That makes sense? In case of lymphomas, for instance, it can be one lymph node. It can be multiple enlarged lymph nodes because, say, if that's B-cell lymphoma, those lymph nodes will keep churning out B-cell dysfunction. Now, going back to function RBCs, red blood cells, okay, when they get destroyed, that's spleen job. Now, remember we talked about malaria, how red blood cells that are sickle-shaped are destroyed. Infected red blood cells are destroyed by spleen. Well, guess what? In actual clinical malaria, all those infected red blood cells will be destroyed by a spleen, and that massive effort on destruction and recycling will lead to splenomegaly. Now, any anatomical features? Yes. So, just as the lymph node, there's a capsule. There are trabeculae that break the spleen down into several compartments. There are two types of pulp: red pulp and the white pulp. Okay, the white pulp is around the arteries, mostly lymphocytes. Red pulp is penetrated by a bunch of splenic sinusoid capillaries. You can see them here, and that's where the red blood cells and macrophages. Red blood cells can be released back in the blood, and macrophages in spleen dispose of the pathogens and old blood cells. That makes sense? So I have two pulps. White blood cells is rich in white pulp is rich in white blood cells. Red pulp is rich in red blood cells. Sorry. End of the day. Other follicular lymphatic tissue. Spleen, thymus. Also two layers, cortex and medulla. Now, thymus is quite interesting. It grows throughout childhood and adolescence. And then in the early adulthood, it starts to shrink. And if you would look at the size of a thymus in, I don't know, 40, 50, 60-year-old person, it's very, very small. But interestingly enough, it still produces immunocompetent T cells. The function of the thymus, as you remember, is the maturation of T cells. Does that make sense? One and only function. Okay? Maturation of T lymphocytes. The hormones thymosin, thymulin, and thymopoietin regulate the T cell maturation. Okay? Doesn't have B cells at all. No B cells in thymus, only T cells, a bit of macrophages. What is interesting, thymus is the school for T cells. Do you understand that? It's the school where T cells learn what is self and what is not. Does that make sense? So, that's the time when T cell basically get exposed to self-antigens, learning what are the self-antigens. This is why thymus is separated from the circulation by the blood-thymus barrier. Why it's so important? Imagine that there is some kind of an antigen that crosses blood-thymus barrier and gets in the thymus. T cells in the thymus are yet immature, so if they encounter some antigens, they will consider those antigens self. You see what I'm saying? Like everything they learn is safe. Okay? So if some kind of a pathogen manages to get into the spleen, into the thymus, T cells will learn that it's, not a pathogen. Really, it's a part of the human body, and will never recognize it. Very insidious but smart strategy. I don't know about any human infections like this, but in mice, it was shown that mouse memory tumor virus, that quite literally causes memory tumors, actually can cross blood-thymus barrier, get in the thymus, and therefore avoid further recognition. Make sense? Now, MALT, mucosa-associated lymphoid tissue, found in the mucosa, in the lamina propria, include tonsils, pear patches, and the appendix. So here you can see tonsils with the follicles and germinal centers in them. Around pharynx, lingual tonsils, palatine tonsils, pharyngeal tonsils, and tubal tonsils. Palatine, oral cavity in the back. Okay, so you can see them, they kind of up there, close to the soft palate. Lingual tonsils is at the back of the tongue. It's really hard to spot them because it's really hard to see the very, very back of the tongue. Pharyngeal tonsils are at the posterior wall of the pharyngeal tube. You can see them too. Tubal tonsils around the auditory tubes, not necessarily can be seen by yourself. Respond to any kind of respiratory infection. Peyer's patches. Lymphoid follicles in the lamina propria of the gut. So small intestine and, very importantly, appendix. Peyer's patches in appendix and in the intestines in general now are considered to play a really important role in autoimmune diseases such as inflammatory bowel disease. They are the focus of the many IBD studies. They basically respond to the pathogens that cross intestinal wall. That make sense?
So the appendix actually does have like a job, and that's what you're saying is it gets the bacteria that will cause IBS, or no?
I would love to give you the answer to your question. So we have a population. I'm going to take about 5 minutes to try to explain why I can't give you an answer. Bear with me. I'm trying to get to anatomy. Okay, here. This gentleman right here. Focus on this gentleman's gut. Can you see that? The gut. Your gut has a lot of normal bacteria. We clear? And these normal bacteria interact with your normal immune system. One of the roles of Peyer's patches is to make sure that your normal immune system does not produce any abnormal immune responses to those bacteria. Make sense? So like immune tolerance. That's one. Second, Peyer's patches have to respond to pathogenic bacteria. Chances are your appendix is not going to see pathogenic bacteria at all because that part of the intestine, it's not where the food is coming through. Does that make sense? What we think appendix is doing, it actually regulates the interaction between your commensal bacteria, the normal bacteria, and your immune system. And it shapes your immune system development. Now, is it linked to IBD? I would bet $100 yes. Because improper maturation of your immune system may contribute to the development of the autoimmune disorder. And if you remove, hypothetically, if you remove appendix at the age of 6 months, just to be on the safe side, that will change the whole immune system development, and that will change the immune responses, and that may lead to autoimmune disorders in IBD. But I would argue that Peyer's patches in the appendix play more role in shaping immune system than in responding to actual pathogens. Did I give you an answer? Oh, okay. It can be microbial abnormality, or it can be pathogenic bacteria that will cause local inflammation. Okay, so local inflammation means enlargement, means pain, and it swelling, stretch, and it can legitimately burst because of the fluid accumulating in the appendix. And if it bursts, then it's a different story. Okay, so it's not because of food is getting stuck in the appendix. I was threatened when I was a kid that if I will eat sunflower seeds without husking them, just, you know, the whole thing, I will get appendicitis. Here I am, with appendix intact. So most likely it's bacterial infections. Questions? Okay, so I thought long and hard